Welcome and thanks to everyone joining us today for the next instalment of Imugene's Science Series, where we go a bit deeper to learn more about the people, history and details behind Imugene's science. Today we're going to focus on Imugene's B-cell activating immunotherapy, PD1-Vaxx, and just noting that it is definitely PD1-Vaxx. There's a bit of confusion with CheckVac being on the title of one of the slides earlier. The technology was developed by Professor Pravin Kaumaya, Director of the Division of Reproductive Biology and Vaccine Research at Ohio State University, who is joining us today. He has a background in immunology and oncology with specialised training in chemical biology centred on cancer immune system interaction. The focus of his research is on directing patient immune systems to recognise and kill their own cancer cells. He's now authored and co-authored more than 130 peer-reviewed articles in well-regarded journals and has either been issued or is pending approximately 40 patents. Joining Professor Kaumaya today, I also want to introduce Imugene's recently appointed Senior Director of Clinical Science, Dr. Nimali Withana, and Imugene's Managing Director and CEO, Leslie Chong. Bear with me. In terms of some quick housekeeping, the initial presentations will last for approximately 20 minutes before we would welcome any questions. If you have a question you'd like to submit, please do so using the Q&A panel within the Zoom app. Kick off proceedings, I'll hand it over to Leslie. Please go ahead.
Well, I'm extremely excited to introduce everyone to our PD1-Vaxx and how it came about. In that, just recently, our recent news is all about combination with atezolizumab, and I'm fortunate to be joined by Dr. Nimali Withana, who also worked on it at Genentech Roche. So we know a lot about the drug, and we are fantastically excited about that combination. PD1-Vaxx was invented by the prolific Professor Pravin Kaumaya, who we are so delighted to be joined by today. So I'm really looking forward to this presentation, and I hope you enjoy it as well.
Okay, thank you, Leslie. I think you went overboard saying I'm prolific, but I'll take that. And can I have the next slide, please? Okay, so if we look at the landscape for immuno-oncology and checkpoint inhibitors, there are a number of monoclonal antibodies, humanized monoclonal antibodies that have been FDA approved. For example, for HER2, you have Herceptin and Perjeta. PD1 is what we're going to be talking about today. And of course, there is Keytruda, which is pembrolizumab, that has been approved by the FDA and is being used in the clinic. And for PD-L1, there are a number of different monoclonal antibodies, and the particular one that I'm referring to is atezolizumab, Tecentriq. So what is our strategy? And of course, with all the monoclonal antibodies, you have to infuse them into the patient. They're very expensive. And one of the problems with monoclonal antibodies is you have to inject it, infuse it together with a chemotherapeutic agent. So is there a better way to make antibodies to treat cancer? And in fact, there are B cells in the body that naturally produce millions of antibodies. And what we've been doing for the last 30 years is to try and teach the B cells to make antibody using peptide antigen by engineering chimeric B cell and promiscuous T cell epitope for a universal vaccine, and we vaccinate with this. Next slide, please. So for the PD1 epitope vaccine, the way we go about doing this is we predict epitopes on the surface of the protein, and in this case, PD1. And we actually do algorithms that are antigenic or immunogenic, and we come up with some epitopes that would be on the surface of the protein. Therefore, we could identify those. And what we then do, next slide, is we engineer those peptides based on crystal structure determination of, for example, PD1 and PDL1, or PD1 and the various monoclonal antibodies. And that helps us to engineer the correct epitope. And this is something that we do quite routinely in my lab. Next slide. So we identified 4 different epitopes for PD1. And what we then do is we look at their structure, and we then do a series of studies. And this particular, next slide, this particular PD1 B cell epitope, now known as PD1-Vaxx, which is actually Imugene vaccine that is in clinical trial. The way, I'm not going to show you all the data that was published in oncoimmunology, but I'm just going to give you one set of data that shows that we can vaccinate mice every 3 weeks so that they generate a good immune response. And once we have done that, we then challenge them with, in this particular case, a colon carcinoma cell line, CD26. And we then monitor tumor growth. And as you can see in panel B, those peptide immunogen are quite immunogenic, and they generate very good isotype of antibodies. In this particular case, there's a combination of IgG1 and IgG2A. And then on panel D, what we show there is the individual mice. There's 10 mice per group. And in panel E, what we show here is that the standard monoclonal antibody that was generated for example, pembrolizumab, we have the mouse monoclonal antibody, and we treat the animals with that. And you can see vaccination with our epitope has very similar profile for inhibiting tumor growth. And if you look at panel F, we show that our PD1-Vaxx has a good CR. Next slide. So how does this vaccine work? So if we take PD1-Vaxx and we combine it with IC720, which is an RLSL oil and water emulsion, we make the emulsion with the peptide with an adjuvant, and then we inject it in the patient. And what happens is, if you look to the right, is our construct doesn't get proteolyzed, and therefore we generate very high affinity polyclonal antibodies. What happens is that the B cell is on the surface, the T cell part of it in orange binds MHC class 2 and activates the appropriate cytokines to help the B cell make the appropriate antibodies, and that then prevent the tumor from growing. Next slide. So just a quick overall strategy from beginning to end. So we predict antigenic epitopes, we model those with 3-dimensional structure, we get a lead design, and then if you look on the right hand side, we generate antibodies in rabbits so that we can do the proper mechanism of action of those antibodies. In other words, we look at proliferation, phosphorylation, immunogenicity, ADCC, apoptosis, and a whole battery of in vitro assays that helps us determine the lead candidate for optimization. And then we go into animal, whether it is transgenic, syngeneic, or transplantable model, we challenge with cancer cell line. We are able to finally pick 1 or 2 epitopes, and we test the efficacy of the combination in mice, and then these could then go into clinical trial. This whole procedure, now we've got it to a T in terms of it takes about 6 months from beginning to end. Next slide. So this is PD1 that I've just talked about, that Imugene has in clinical trial, and I think Nimali will be talking about that. So we now have also designed in a very similar fashion PDL1. Of course, PD1 and PDL1, they are interact. And so we have now again identified a number of epitopes that we do the same procedure of vaccination and mechanism of action. And as you can see here, all 4 epitopes are highly immunogenic in rabbits. Next slide. So we then go into mice again, and we take all the 4 epitopes individually, vaccinate mice at 3 weeks interval, then challenge those in this particular case with the CD26 carcinoma cell line. You can see panel B, high immunogenicity of the different vaccine. And then we look, of course, for any antibodies, they have to interact with the native protein. And in this case, when we look at panel C, you can see all the antibodies generate good response to the native protein. And then on panel D, we look at tumor growth. And as you can see here, compared to the negative control, most of our epitopes have been shown to show inhibition of tumor growth, but the PD-L1 130 and 36 is the prime target to move forward. Next slide. And let me tell you that this particular set of data hasn't been published yet. We are preparing the manuscript to submit. And what we also then did was we looked at not only syngeneic bulb C mice, but we look at different cancer model, in this case, mammary cell lines, D2F2 and 41, which is TNBC. And as you can see here, in panel B, highly immunogenic, generating a maximum of IgG1 antibodies. And if you look at panel D, E, and F, D and E, you will see that our vaccine 130 is much better in the D2F2, equally good in 41 in inhibiting tumor growth. And when you look at panel F, you will see that our MVF PD-L1 is highly relevant for moving forward as another potential vaccine. Next slide. Okay, and again here we did we looked at CD26 mammary tumors and 41, and we looked at in a different, we made a peptide now instead of our measles virus, we use tetanus toxoid, and we can then look at the immune response. And essentially, if you look at all those data, it shows that our PD-L1 130 is very good and inhibits tumor growth in all 3 mammary and colon carcinoma cell lines. Next slide. And again, here, this is in another set, and it's always good to do different cell line, cancer cell line. MC38 is colon, and B16F10 is another good cell line to look. And in fact, if you look, high immunogenicity in B, and in fact, all our vaccination procedure is to vaccinate every 3 weeks and then challenge after 2 weeks after the last one. And if you look at panel D, E, and F, you can see again here at the PD-L1 in all 3 carcinoma cell line very, very good in terms of inhibition of tumor growth. Next slide. Okay, so one of the major problems in checkpoint inhibitors is mechanism of resistance, as it is with targeted therapy. So therefore, if you are resistant to PD-1 treatment with Nivo or Pembro, then you get upregulation of LAG3 or TIM3. And this is where we are going. And of course, if you look at CTLA4, for example, ipilimumab is one agent that is being used in the clinic for PD-1. And then if you look at LAG-T, TIM3, and TIGIT, there are a number of other agents that is in clinical trial, not quite approved. So what is it important for us then is to come up with new vaccine as opposed to human monoclonal antibody. Next slide. And in this particular case, we have just completed designing and testing a CTLA4 peptide epitope. And I'm only referring to those epitopes as P1, P2, and P3 and P4 because we haven't done the patent yet. But if you look again in our similar model, looking at CT2641 and D2F2 a cell line, you can see we have two epitopes that we have identified as P1 and P4 that we could then move those forward for combination immunotherapy together with our PD-L1 or PD-1. Next slide. Okay, so these are studies that are ongoing now to look at combination. So we're looking at V29, where we're doing PD-1 and PD-L1 together, V30 CTLA4 and PD-L1, V31 PD-1 and PD-L1, and V32 CTLA4 peptide mimics. And our control in there would be to use the humanized monoclonal antibody, or at least the mouse relevant monoclonal antibody to PD-1, PD-L1, and CTLA4. Next slide. Okay, so Imugene has licensed most of our portfolio, and I'm just showing here what we are presently doing. Of course, we have the combination of PD-1 and HER2. We have the PD-L1 vaccine, which is in process, combination of PD-L1 with other targeted therapies. We have CTLA4, TIGIT, LAG3, and TIM3. So TIGIT is now in process. CTLA4 and PD-L1, we already know which epitope to go with. LAG3 and TIM3 over the next year will come up with their epitope that we could then use in combination, which is where we will be going. Combination immunotherapy with checkpoint inhibitor as to be the future of immuno-oncology. Next slide. Okay, so thank you for your attention, and we'll move on to the next speaker.
Lovely. Thank you, Professor Pravin Kumar. You explained that eloquently and you introduced the science as to why we're doing what we're doing. So what I'd like to do now is just shift gears a little bit and talk about why PD-1 and PD-L1 combination, why we're interested in this, and how does PD1-Vaxx play a role in here? What we know is that those patients who are treated with PD-1 and PD-L1 alone do have phenomenal responses. And we've seen this in lung cancer, in breast, in prostate cancer. But there is a portion of patients who do not respond. And what we have come to understand is that it's really critical that we look at combination therapies because it's critical to hit both pathways. And what we've observed in preclinical and clinical data is what you can see happen when you've got both PD-1 and PD-L1 targeted. So we look at pancreatic cancer as an example in the top panel. You can see here that those that are treated with just the individual PD-1 and PD-L1 do respond compared to the control. But the true benefit actually comes when you've got that dual targeted therapy. And we also see this in melanoma, where when you've got that combination, it actually prolongs survival. We know that there are other pharmaceutical companies such as Eli Lilly and BeiGene that are also looking at this combination. Eli Lilly, for example, have a bispecific monoclonal antibody that targets both PD-1 and PD-L1. And with BeiGene, they've got an individual one where they're looking at PD-1 and PD-L1. We're seeing efficacy results from there, and we know that there's a benefit there. So this got us excited and as to why we're moving forward. So if I go on to the next slide, I want to introduce our study, the one that we're really proud of, the Imprinter study. This is where we've used Professor Kaumaya's incredible PD1-Vaxx. And we're going to be looking at these patients who have advanced metastatic non-small cell lung cancer. We know these patients have incredibly poor outcomes, and it's actually one of the leading causes of deaths, cancer deaths around the world. So this has the potential to make a big impact for our patients. We wanted to start out by looking at the monotherapy. So what happens when you use PD1-Vaxx alone? We're looking at 3 different doses in the first part. So this is doses starting from 10 micrograms, 50 micrograms, and 100 micrograms. And I want you to keep in mind that the patients that we enrolled into this part of the study, they had actually progressed on prior immunocancer therapy. So what we've observed, and I think you might have heard about this earlier, is that we've actually observed one patient who's had a complete response. And in fact, this patient's actually been on 12 months of treatment and is doing really well. As physicians, as researchers and drug developers, these are things that we strive for. And it's when you hear these outcomes, it's what gives us hope that we're on the right path. To know that we've got a drug here, PD1-Vaxx, that actually has very low toxicities, and to see this kind of response is really encouraging for us. So as I mentioned, we've just cleared the monotherapy where we've looked at these 3 cohorts, and now we're just about to open up into the combination. Leslie alluded to this before. It's a combination that we're very proud of. We've both had partnerships at Genentech and Roche with this molecule, atezolizumab. We've seen tremendous impact that this drug can have. And what we're excited about, and both what Roche is excited about, is what does this combination hold for the potential for these patients? So in this second part, we've actually got 2 arms. The first arm will enlist and enroll patients who are treatment naive to prior immunotherapy. And then the second arm is at those patients who have progressed on prior treatment. We'll be looking at two cohorts here. The first one is looking at PD1-Vaxx at 10 micrograms in combination with atezolizumab at 840. This will be dosed every 2 weeks. The second cohort is 50 micrograms of PD1-Vaxx in combination with atezolizumab. And based on that, that will allow us to determine what we want to do with the combination moving forward. As I mentioned, this is something incredibly exciting for us and something that we're really looking forward to. And what I'd like to do next is just show you on a timeline where we're at. So next slide, please. So this is the current status. We've just cleared those cohorts that I mentioned, and we're currently looking at the safety and the profile and the responses that we see to help us determine how we move forward with that combination study. The study could not go ahead without these incredible partnerships that we have with our sites. These investigators are incredibly enthusiastic about this combination. And in fact, when you do hear about these kind of responses, it encourages you as to why we're moving forward. So a massive thank you to our sites, both in Australia and the US, that really pushed and helped us move this forward. So we're excited and we can't wait to see what the next phase brings forward, but it's early days and we're really looking forward to what happens next.
Thanks so much, Nimali. I just want to have one minor correction. That patient that had complete response is actually on his 14th, I dare say almost 15 months of clearance from his tumor burden. So spectacular for that patient.
Absolutely spectacular. Yes. Thank you, Leslie.
Thanks, Leslie, and thanks to Dr. Withana and Professor Kaumaya for those insights. We'll now move on to the Q&A segment. Once again, if you do have a live question you'd like to submit, please do so using the Q&A panel within Zoom. But the first question I'll direct to Pravin, which is, tell us how you met the team at Imugene and when you first met the team.
Thank you, Matt. So I happen to have given a talk in TIDES Europe in Vienna in November of 2017, and it was to do with my entire portfolio, but included PD1. And what happened was that Nick Eady saw the abstract and contacted TIDES Europe to ask whether he could have a set of my slides. But they referred him to me, and Nick sent me an email, and I've known about him and his group in Australia. So I gave him a call and FaceTime, and he blurred his, I think he was still in his pajamas. But anyway, so we had a chat for about an hour, and I am fully aware of Imugene and the work that's been done in Vienna. And so I was interested in what they had in mind. So I sent Nick all my PowerPoint presentation. And the next thing I knew, he called and said, he and Leslie were going to show up in Columbus, Ohio. So I said, okay, we'll wait for you to come and discuss more. And lo and behold, Leslie and Nick showed up in Columbus, and we went over my entire portfolio, and that's how I got to know Imugene. And of course, we've licensed some of my previous portfolio to some companies in the US, but they were not very good companies, and we had to extract our license back. But having looked at Imugene, their portfolio and their, how they had brought their Vaxx and B-cell vaccine. So I was really very, very keen on combining what we're doing together with Imugene.
Great, thanks for that, Pravin. The next question is another one for you, which is, how does PD1-Vaxx differentiate from the billion-dollar blockbuster drug Keytruda from Merck?
Right, so that's a major difference between what we do. What we do is we map epitopes on PD1 or PDL1 or CTLA4. We have small peptides that we combine with a promiscuous T cell epitope. So our vaccine is about 50 amino acid in length, and you have the patient make the antibodies. So it is very, very safe. As opposed to using, for example, the monoclonal antibody, humanized monoclonal antibody that you infuse together with chemotherapeutic agent. Our vaccine is naked, just our vaccine that creates the antibodies very similar to Pembro or nivolumab. And so it's the patient that generates those antibodies, and in so doing, it is very, very safe.
Great. And another question, Imugene is combining PD1-Vaxx with Tecentriq, as you know. Tell us more about PD-L1-Vaxx and how it compares with PD-L1 monoclonal antibodies.
Right. So as you've seen in my set of slides that we haven't published yet for the PD-L1, our PD-L1 vaccine 130 is not only comparable, but better than the mouse monoclonal antibody that represent atezolizumab. So in other words, our vaccine is much, much better in terms of inhibiting tumor growth in various syngeneic models that have been challenged with various cancer cell lines. And as you've seen in my presentation, we've done at least 3 or 4 different cell lines, different cancers, different syngeneic model, and we are quite confident that our PD-L1 vaccine would be equally effective if we were to combine it with our PD1. And in fact, those studies are now in progress in the lab.
Great. And you've touched on it a little bit already, but what excites you the most about the new checkpoint inhibitor vaccines you're developing with Imugene, and will combinations be the key to this, do you think?
Yeah, and combination, we've been doing combination immunotherapy for the last 10 years by using HER2 and VEGF and IGF and so on, and we've published extensively on that. So I am excited about getting, we have a PD1 and a PDL1, we will have a CTLA4, and we're looking at TIGIT, TIM3, and LAG3. So all those preclinical studies in syngeneic model are going to be done over the next year, and I'm pretty sure we will obtain a combination of either one or the other that would be extraordinary in inhibiting tumor growth. So I'm excited about that. We have spent some 30 years developing our vaccines on B-cell vaccines, and we are now at the stage where we can take them forward. And with Imugene, obviously, who is funding a lot of the work being done in my lab, is to move forward into other clinical trial with the best possible combination.
Thank you for that. Nimali, a question for you now. What attracted you to joining Imugene?
Oh, great question, Matt. Look, honestly, it actually is that innovative platform that we've got. The moment I saw that, I realized that we have the potential here to make a huge impact with our platforms to our patients. So when I got talking to Leslie, I immediately knew that this company is doing something that is going to make a huge difference, and I wanted to be a part of it. That was the first thing that drew me, but I have to say very closely followed by the incredible team that is part of Imugene. I have the opportunity every morning to wake up and work with a team that's incredibly smart, they're driven, and really what holds me is the fact that we're so passionate about the impact that we can bring to our patients' lives.
Right, and another one is, what do you think? What do you think differentiates Imugene from other biotech companies from your experience?
Great question, Matt. Look, I think what Imugene has got is actually something very unique. We've got these incredible shareholders. It's not just those that invest in the company that we have these close relationships with, but we have these incredible relationships with our inventors. I mean, Pravin Kumar is just an example here. He's an inspiration to see the work that they do. And we have these incredible relationships with them. And also the impacts that we have with our sites and the patients, and knowing that we have the ability to bring all of this together. So it's not just those who invest in our company, our inventors, our partners, but we're really making a difference here and trying to push that needle and trying to make a difference in people's lives. And that's how I think Imugene truly stands out. They collaborate.
Great. Look, that's all the questions that have come through. I want to give a big thanks to Professor Kaumaya for taking the time out of his busy schedule to present for us today, as well as thanking everyone who joined us for this instalment of Imugene's Science Series event. We are looking to set up another one in the not too distant future, so please stay tuned for details on that. But with that said, we will leave it there for today, and thanks again to Leslie and Professor Kaumaya and Dr. Withana for joining us.
Thanks, everyone.
Thanks, everyone. Bye for now.
Bye.
Bye.